Hello. Last lecture we finished with trying to describe the different forms of abnormalities in sperms, and we understood that in order to be normal male, it had to have in the ejaculate around 200 to 300 million sperms. 60% of these sperms must have a normal shape, and 40% must show vigorous motility. So let's talk now about the oogenesis in mammals. So to understand this figure, we need to correspond each stage during the meiosis of the during oogenesis, which with its corresponding location either in the ovary or the oviduct or location. So you can see there is a germinal cell. Let's have a pointer. So you can see there is a germinal cell, which usually these germinal cells considered to be and form the primary follicle, and you can see the location of this primary follicle, and in that stage, the cell is diploid. Once there is increase in the size of the developing follicle, soon after that, it will form a mature follicle with a secondary oocyte, and you can see the secondary oocyte. And actually, the secondary oocyte, which is now haploid, it is a stage where the follicle got ruptured and released into the cavity in which it got collected by the fallopian tube or the oviduct. Once it moves in the oviduct in the stage of secondary oocyte, it waits for the sperm to be fertilized. And you need to understand that in order to have an efficient fertilization, the secondary oocyte must be in contact with the sperm to form the ovum at the first third of the fallopian tube. So in case the sperm meets the ovum here, this will lead to some complication in the process of fertilization. So why it's important to have the fertilization in the first third of the fallopian tube? To give some time during traveling all over the fallopian tube before entering the uterus, in order to do the cleavage process, which is a following stage after the process of fertilization. يبقى اللي عايزين نقوله ايه؟ ان عمليه التلقيح لازم تحصل في الثلث الاولاني من قناه من فالوبيان تيوب. خلاص عشان تدي فرصه للامبريو اللي هيتكون ان يعمل عمليه الكليفج بتاعته او الانقسامات الكتيره قبل ما يحصل له عمليه الامبلانتيشن في اليوترس. اوكي؟ So again, here we will see some simple differentiation between spermatogenesis and oogenesis. Of course, location is different based on the type of the gametogenesis, and as well the number of gamete produced. So normally in female, it is fixed amount, usually around 400 ova are formed. But of course, in male, it's millions and lifelong production. Gametes, of course, are 4 in spermatogenesis, 1 in oogenesis. The beginning of process starts at puberty in spermatogenesis, while it begins during fetal development for the oogenesis. The timing of gamete continues anytime throughout the life, but the timing of the gamete for the female is once a month. The end of the process, as we said, the fertility is lifetime, but the efficiency is reduced, while the fertility stopped at the menopause. The timing of gamete release is any time, but of course for female, it is monthly a cycle. The meiotic division is uninterrupted, but as we said before, it is arrested in case of female until it is fertilized by sperm. The germline epithelium. So you can see in spermatogenesis, the germline is involved in the process of gamete formation, while germline epithelium. What do we mean by germline epithelium? Because it's not involved in gamete production. If we go back to this figure, you can see that the starting material of the oogenesis is outside the epithelium, which is represented here by two layers, two lines. But in case in the spermatogenesis, you can see that the epithelium, which is the starting layer, is involved in the process of the spermatogenesis. Any questions? Okay. Let's see now how is the behavior of chromosomes during meiosis, and we'll take for examples, as you can see, you can see here four chromosomes, as you can see. So in the process of interphase, you can see that there is homologous chromosomes. To understand, these are the DNA contents. So you have two homologous chromosomes. Two pairs of homologous chromosomes, the red and the blue. And then in prophase 1, of course, there is starting to align, and the process of, during meiosis 1, the process of recombination, exchange of segments between the chromosomes. So you can see that the process of reduction division, instead of having 4, now it's having 2 in each. And soon after that, soon after that, the reduction division occur, and you can see that the daughter nuclei now occupy one copy of each chromosome, and already the recombination occur. So let's see here how to refresh your memory, how the process of the behavior of chromosomes during meiosis. In this animated figure, you can see that homologous pair of chromosomes align to the equator, and after recombination, the spindle fiber contract, and each now is said to be to one side of the cell. The nuclear division, represented by kinases, is followed by cytokinesis. In cytokinesis, the two cells are separated, each with its own chromosome. That soon the process is repeated, but in this case, it's like meiotic division. So you can see now 4 haploid cells are formed. Again, you can see the animated figure. طبعا السلايد دي بتفكركم بعمليه الميوزس عموما، سواء كان بقى سبرماتوجينيسيس او اوجينيسيس. وكلنا عارفين ان whatever في السبرماتوجينيسيس or oogenesis, the end product is 4 cells. But in case of males, the end product are 4 sperms. Like in case of females, it's only 1 ovum and the 3 polar body. يبقى حد يقدر يقول لي ليه في الفيميل احنا بيطلع لنا 3 polar bodies؟ حد يقدر يقول لي؟ لسببين. حد عارف السببين؟
مش ويساد انه الاج بيكون بدها كل النيوترينتس وبتاخد كل شيء سيتوبلازم كرمال.
يبقى اول سبب تو كونسنتريت. يبقى اول صح يا ابراهيم. يبقى اول سبب تو كونسنتريت ذا سيتوبلازم ان جاست وان سيل، صح كده؟ تمام؟ وبالتالي السيتوبلازم كونتين نيوترينتس، ده اول سبب. ثاني سبب مهم، حد فاكر؟
كرمال انه يصيروا هابلويد.
ممتاز. يبقى السبب الثاني عشان يبقوا هابلويد احنا محتاجين الميوتك ديفيجن، صح كده؟ اوكي نكمل. So for gametogenesis in general in mammals, in female, meiosis, as we said, begins in the embryonic bones, while in males, meiosis is not initiated until puberty. So this critical difference in timing is due to a very important molecule, which is the retinoic acid, which is produced by the kidneys. So the embryo of the fetus inside the uterus have the kidney which can secrete retinoic acid. And now we'll see how this retinoic acid is very important in deciding the type of the gonads. So this retinoic acid will stimulate germ cells to undergrow to undergo a new round of DNA replication and initiate meiosis. In males, however, the embryonic testes secrete the retinoic degrading enzyme, which is called CYP26B1. So this degrading enzyme, this RNA, retinoic acid degrading enzymes, will stop the occurrence of any meiosis. And that's why the meiosis do not start in the gonads of male fetus until puberty, okay? Because retinoic acid stimulate DNA replication, initiate meiosis in male. So because it's not secreted in the fetus of males, that's why the spermatogenesis start after being secretion of the retinoic acid, which is until puberty stage. So let's have a very good figure describing how the number of germ cells in the human ovary changes over lifespan. So initially you can see the y-axis represents the number of germ cells times mean, and you can see the x-axis represent the month before conception. So you can see that the number of germ cells increase up to reaching around the 5th month. Soon after that, it will decrease in the number. We are talking now about the number of germ cells, not the number of the ova. The number of germ cells mean the initial precursor, which is called oogonia. Oogonia is the plural, oogonium is the singular. So they reach a certain number just before birth. After birth, it soon decrease in number until age until the 15th month, and it's obvious that is almost decreasing, slight decreasing, because as you know, the female loses ova during the process of the menstruation. So now we understood the how the process of spermatogenesis occur, how the process of oogenesis occur, and how the ovum is produced and the sperm is produced, and as well we understood the structure of both of these gametes. So now the stage is set for fertilization. We have two gametes ready for fertilization to take place. The egg and the sperm now will both die if they don't meet. يبقى بعد ما تكون الاثنين دول لو ما حصلش fertilization هيموتوا الاثنين ما لهمش لازمه. The conclusion of that part or coda, the elements, these elements, which is the sperm and ova, that unite, each of them are single cells, each on the point of death, because if there is no fertilization, both of these cells, gametes, will die. But their union, the union of each of these single cells, can rejuvenate and a new individual is formed, because there is a zygote will be formed, and this will constitute a link in the eternal process of life, and this provide the eternity for the human species. خلاص؟ في حد عنده سؤال لغايه دلوقتي يا شباب؟
دكتور شو يعني كودا؟
كده يعني conclusion باللاتيني. اوكي. يعني عندنا سبيرم واوفا هيموتوا لو ما حصلش فيرتلايزيشن. شوفوا مع انهم هيموتوا لو ما حصلش فيرتلايزيشن، لكن لو حصل فيرتلايزيشن الاثنين مع بعض بيدوا individual كامل بيقدر يعيش. اوكي. So now we move to the next topic, which is fertilization. And actually, fertilization, it is the beginning of a new organism. So as we all know, fertilization is process of two sex cells, two gametes, fused together to create an individual with genetic potential derived from both parents. Okay. So the fertilization have to accomplish two separate events. to transmit genes from parents to offspring, obviously, and to initiate reaction for the egg cytoplasm to permit development to proceed. يبقى ده الهدف الرئيسي من الفيرتلايزيشن، ان انا انقل الجينز اللي موجوده افتر طبعا الكومباينيشن اللي حصل في الميوزز وكل الكلام ده كله، انقل الجينز دي للاوف سبرينج. وتاني حاجه ان الاوفم once is fertilized and converted to a zygote لازم يكون عندها الميكانيزم to initiate development، لازم يحصل لها cleavage عشان تقدر تكمل وتدينا embryo. So generally fertilization consists of 4 major events. اول حاجه sperm and egg get contact and recognition. لازم السبرم يتعرف على الاوفر بتاعته، ودي حاجه من الحاجات المهمه جدا يا جماعه، عشان لو ما حصلش الكلام دوت وما حصلش فيه سبيسيفيكيشن في التعرف ما بين السبرم وإيتش سبيسيفيك ايج، ده هيؤدي لعدم توازن ولخبطه في الاورجانيزمز. ان لو السبرم يقدر يتعرف اي ايج فبالتالي يعمل لها فيرتلايزيشن فهيطلع لنا كائنات غريبه. خلاص، فبالتالي في ميكانيزم بتربط كل سبرم بإيتش سبيسيفيك ايج. فمهم قوي يحصل كونتاكت، وبعد الكونتاكت لازم يحصل ريكوجنيشن للاوفر. The second major event is the regulation of sperm entry into the egg. لازم يبقى في mechanism that ensures only one sperm can fertilize the egg. ثالث event بعد ما خلاص دخل السبرم internalized inside the ova, the genetic material must be fused. The haploid genetic material must be fused. The haploid material of the sperm and the ovum. The last event, once حصل fusion and now the zygote is 2n، حصل له restoration تاني للديبلويد number في genetic material، لازم يبقى في activation of this zygote or this fertilized egg to start cleavage, to start multiplication, to start development. So majorly these are the 4 events that we will talk in the topic of fertilization. So let's talk now about the recognition of egg and sperm. Initially, the interaction of the sperm and the egg generally proceed according to 5 basic steps. يبقى أول event اللي هو عملية الريكوجنيشن للإج والسبرم ده نفسه بيتقسم ل 5 basic steps. أول حاجة بيحصل chemo attraction. What's meaning of chemo attraction? That the sperm is attracted to the egg by the help of a chemical secreted from the egg to attract its own specific sperm. Okay. Then there is a process of exocytosis of the sperm. So the sperm, as we all know, have an acrosomal vesicles which contain a lot of proteolytic enzymes, and a lot of enzymes that need to degrade the wall of the egg. So there is exocytosis. The sperm release these enzymes once in contact with the egg in order to degrade the cell membrane. After that, it will bind, the sperm will bind to the extracellular matrix, which is a layer around the egg. It's either called the vitelline envelope or a zona baliasta, and we'll see later in the coming few slides the difference between them. The 4th step is once there is a contact, a recognition, a lysis of the cell membrane, that now the sperm is ready to pass through the extracellular matrix and then fuse the egg and cell membrane in order to have the genetic material fused inside the egg. So let's see here a summary of the events that lead eventually to the fusion of the egg and sperm membrane in sea ocean. And we are taking an example of sea ocean as an example of external fertilization. What's the meaning of external fertilization? The sea ocean, حد عارف السي ocean اللي هو ايه يا شباب؟
يس هيدا بحطوه كمان مع السوشي I think.
طب اسمه ايه بالعربي عارفه؟ حد سمع عن حاجه اسمها توتيا؟ ها؟
No.
Okay, it's a kind of a sea star. هو شكل السي ستار يبقى ساعات بيقولوا عليه انفض البحر اللي هو له شوك كده طالع كتير جدا. اوكي. خلاص؟ طب ايه اللي بيحصل؟ What is this external fertilization? Usually the male organism, the female organism will lay eggs on top of the floor of the sea outside her body, and then the male sea ocean will discharge sperms outside of his body on top of the egg. So basically, the process of fertilization occurs outside both male and the female. So that's why it's called external fertilization. So now we see the steps of this fertilization in sea ocean. So you can see here, but keep in mind that these fertilization occur in sea water. That's why it's called external fertilization. So now the sperm, you will see that this is a part of the egg. You can see this, let's have a pointer. This part of the egg, all this is part of the egg wall. So you can see the egg cell membrane is that region, the egg cell membrane. This is followed by vitelline envelope, which is the pink layer here. And then you have a very protective layer to the egg is called jelly coat, which is the extracellular coat. So the problem now for the sperm, it needs to contact, it needs to reach this egg cell membrane to penetrate inside. So it has many obstacles. It has the jelly coat or the jelly layer, then it has the vitelline envelope. So you can see the structure of sperm is typical here. The sperm start to contract. You can see it start to contact first the jelly layer. Soon after contacting the jelly layer, you can see extension of the acrosomal process, and it swims its way through the jelly layer. And this will be helped with the acrosome reaction, and you can see some of the secretion of the acrosomal region, helping the sperm to penetrate toward the jelly coat and toward the cell membrane. During its movement, it's actually the enzyme secreted from the acrosomal region. It will digest the jelly layer until reaching the vitelline envelope. Here starts the process of binding. You can see that there is binding here between the tip of the sperm, the extended region of the acrosome, with the vitelline envelope. Soon after binding, you can see that there is a fusion between the cell membrane or the acrosomal process, which actually the covering of the sperm, with the cell membrane of the egg. You can see here. And now it's ready to internalize the nucleus to inside of the egg cell membrane, to inside of the egg. فهمتوا كده يا شباب ولا فيها مشكلة؟
تمام دكتور.
Okay. So this slide describes actually the process of fertilization. And you need to understand that this is actually an example of external fertilization. Now we'll take the example of internal fertilization, but generally the difference is so little, but the difference only in the location of the fertilization. So in internal fertilization, the sperm fertilizes the ovum inside the body of the female, and this is the major difference between the internal fertilization and external fertilization. So let's see the process for the internal fertilization. As an example, the mouse. So you can see here, it starts the same as well. The sperm is a little bit different. The sperm is activated by female reproductive tract. So this one of the major difference from the external fertilization, because actually the sperm in the internal fertilization actually moves in the fallopian tube. And once it's moving in the fallopian tube, it got activated by secretion from the fallopian tube. So actually, fallopian tube, it doesn't represent a tube for moving of the sperm until reaching the ovum. It represents a way to activate the sperm to reach the ovum. So you can see here, the layers are a little bit different. You have a zona pellucida that covers the external membrane, extracellular matrix, and then you have the extracellular coat, which is called the cumulus layer. Again, it passes, the sperm passes the extracellular coat until contact with the zona pellucida. Once in contact, there is acrosomal reaction trying to dissolve the zona pellucida. You can see a major difference here, that the thickness of the zona pellucida is much greater than the thickness in the previous slide of the what is a parallel layer, which is called the vital line envelope. So here, the contact, as you can see, lead to starting off the lysis process of the acrosome reaction. Now the sperm succeed in lysing a hole in this zona pellucida and migrate all the way until reaching to the egg cell membrane. And once it reach and contact the egg cell membrane, there is a fusion between the egg and sperm membranes in order to internalize the nucleus to the inside, to internalize the haploid nucleus to the inside. شباب دي فيها مشكلة الجزء دوت؟
No.
Okay, great. So, as we said, that in external fertilization, all the process of fertilization occur in the seawater. So the sea ocean sperm attraction, there is action that occur at distance. So sperms are attracted toward the egg, as we said before, by a process called chemotaxis or chemoattraction. So a chemical can be secreted by the egg. Scientists discovered the chemical and they named it RESAC. They named this chemical RESAC. And you can see here how they managed to succeed in knowing that. So here they done an in vitro in a petri dish experiment. So they have sperms inside the petri dish, and sperms, when there is no egg, when there is no egg, these sperms start to circulate in a circle. That's why each sperm appears as a circle. السبرمز دي يا جماعه في عدم وجود البويضه وفي عدم وجود كيميكال بيوجهها، السبرمز بتبدا تلف على حوالين بعضها فيبان لكم على شكل سيركل. يعني كل سيركل من دي هو يمثل سبرم. ده في البيتري ديش بره في الطبق. طب عملوا ايه العلماء؟ In this petri dish they injected in this solution الريساكت اللي هي الكيموتاكتيك موليكيول ديت بتركيز حوالي 10 nanomolar. Once it is secreted, and you need to understand it is secreted in that location. It is secreted here. So when it starts secreting, you can see over time, this is 1 second. This after 20 seconds of incubating this resac with the sperms, you can see that most of the sperms now are directed, are all directed to the point where the resac material have been injected inside the solution. This gave the conclusion that resac is a very important chemotactic compound that enables the sperm to localize the ova or the egg, because resac actually is secreted by the egg. Because in the sea, the sperm cannot visualize the egg. It needs to sense a chemical compound. So once the egg secrete this resac, it will attract sperms of the same species. So you need to understand that every species, for its egg, secrete a specific chemical compound to attract a specific sperm. So resac actually is a 14 amino acid chemotactic peptide, small protein, very small protein. It was generally the scientists managed to isolate that from the egg jelly coat of the sea urchin, A. panictiolata. And this resac is very specific, and it doesn't attract the sperm of other species, because this resac diffuse from egg The more resac is diffused from the egg, the more sperms are recruited. As well, resac act as a sperm activating peptide. So it caused dramatic change and immediate increase in the mitochondrial respiration and sperm motility. يعني عشان نفسر الحته دي، الكيميكال ده الريساك ده اللي بيتفرز من الايج مش له وظيفه بس ان هو يدل السبرم على طريق الايج، لا ده بيحفزه بطريقه معينه لما السبرم بيحس بيه ان يعلي من الميتوكوندريال اكتيفيتي، فبالتالي يحرق طاقه كتير، فبالتالي يجري بسرعه ناحيه السبرم. شباب فهمتوا تجربه استخدام الماده زي الريساكت ده ان قدرت تثبت لنا ان مهمه جدا اللي تخلي السبرم يتعرف على الاوفر بتاعته ولا لا، السبيسيفيك اوفر ولا لا؟ تمام؟
يس دكتور مش الحال.
طب تقدروا تقولوا لي ايه الفانكشن بتاعت الماده دي اللي انتوا فهمتوه؟
انه تأتراكت السبرمز للأج.
تاني وايه تاني؟ هما 2 فانكشن، وايه الفانكشن الثانيه؟
activate sperm.
عشان يجري بسرعه، صح كده ولا لا؟
يس.
تمام، خلاص نكمل. Okay. Okay, let's continue our explanation of the way that these kind of chemical, the resect, which provide and ensure that each sperm will fertilize its own specific egg. Okay, so generally you can see the egg in the middle. Let's have a pointer. So generally you can see an egg in the middle. So the egg starts to secrete resac, and you can see the gradient of the resac, concentration around the egg. And you can see that the sperm is, initially it was diverted, and then once it senses the resac, actually resac is not a layer, it's a chemical that is dissolved around the egg. So once it recognizes the resac, it starts to migrate toward the egg. But you need to understand here that not only one sperm migrates toward the egg. You can find that all the sperms will migrate toward the egg. So the resac solved the problem of the identification of the sperm to the egg. But still there is another problem, that the egg have to be fertilized by just one sperm. So basically the egg بتلم حواليها السبرم، البنت بتلم حواليها كل الرجاله، وهي في الاخر بتختار ايه؟ راجل واحد. فهمتوا يا شباب ولا لا؟
Yes.
Okay. So now we solved the problem of identification of the sperm for its own egg. Now we can see here is the sperm, how the molecular events occur. How the molecular event once the sperm is recruited toward the egg. So here you can see a representative of the cell membrane of the sperm. You can see you have a calcium ion transporter that permit the internalization of calcium ions, which is very important in the process of recognition between sperm and ova. And you can see as well one of the important receptors on the surface of the cell membrane is called resac receptors, RGC. And obviously, because it's named resac receptor, so it's supposed only to bind to resac. And of course, this resac receptor for each egg is very specific to its own sperm. So once this resac, you can see this is, this is actually the membrane of the sperm, the membrane of the sperm. And this membrane of the sperm have the calcium ion importer and have this resac. So how the sperm recognize the resac? Because this receptor on the cell surface of the sperm will recognize resac in the solution, in the seawater. Once a binding occurs between resac and the receptor on the cell membrane of the sperm, this will initiate the conversion of a GTP molecule into cyclic AMP. The presence of high amount of cyclic AMP will activate the calcium importer to internalize calcium inside the sperm. Once the calcium is internalized into the sperm, this will lead to the activation of the mitochondrial machinery, and it will turn on swimming of the sperm toward the egg. So, as we said, the binding of the resac to the receptor on the cell surface of the sperm will initiate a cascade of reaction in the cytoplasm of the sperm that lead to the influx of calcium inside the sperm. And this calcium is very crucial and very important in regulating the flagellar beats and swimming by this activating sperm through the activating the mitochondrial machinery and the mitochondrial ATP synthase and the ATPases break down ATP to release energy sufficient enough for the process of swimming of the sperm toward the egg. A good experiment has been shown, as you can see. Scientists try to measure and to understand the importance of calcium and to understand if the calcium really is internalized inside the sperm after the process of recognition between the receptor of the resac and the resac molecule. So they measured the calcium levels in this sea ocean sperm after being exposed to resac. So once as we know from the previous slide, once the sperm is exposed to resac, the calcium starts to internalize from the seawater to the inside of the cytoplasm. So in this figure, you can see that the signal of the calcium represented by the coloring, and you can see once the color increase inside the head of the sperm, that means that there is a lot of calcium have been internalized. So basically, it's a very simple experiment. They labeled the calcium, and once the calcium is internalized, they can take an image which show the labeling of the calcium and will show its localization. You need to understand, once it reached red light, once the color is red, as you can see in the last figure, the color red in the middle, that means indicates this is the high level of calcium inside the sperm. If the color is still blue, that means lowest level. So you can see in just 1 second, the level of the calcium inside of the head of the sperm is very high, because you need to understand that calcium is important in initiating the activity of the mitochondria, and hence initiate the breaking down of the ATP to release energy to help in the process of swimming of the sperm toward the egg. So thus, to summarize this point, upon meeting resac, when the sperm meet the resac secreted from the egg, the Arabicia sperm, and this another name for another species for the sea ocean, the Arabicia sperm are instructed where to go and are given the motive force to get there. Okay? Got it يا شباب؟ في حد عنده سؤال؟ شباب صبايا.
تمام دكتور.
بانت هنا أهمية الكالسيوم. احنا لسه المعلومة اللي انتوا لازم تعرفوها ان الكالسيوم هو الانيشيتور اللي بيخلي الميتوكوندريا تستارت اكتف. فهمتوا الفكرة؟
يس.
طب الكالسيوم دخل ازاي جوه السبيرم؟ نتيجه لاتحاد الريساك على الريسبتور اللي موجود على السيل ممبرين بتاع السبيرم. اوكي؟
يس.
اوكي. في حد عنده اي سؤال يا شباب؟
كله واضح.
اوكي. So this conclude our lecture for today, and do you need any questions? Okay. Okay. Thank you and see you next week. شكرا يا شباب.
Thank you دكتور.
يا
باي
شباب.
باي.
باي باي.
ثانك يو دكتور، يعطيك العافية.
شكرا جزيلا. باي باي.